Hey everyone! Welcome to the Ncert Audiobooks Library, the best place to listen and learn from Ncert textbooks. Whether you are preparing for exams, revising a topic, or just curious about something, you can find the audio version of any Ncert textbook here.
2.3 Double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, PEN, figure 2.13a. As this involves the fusion of three haploid nuclei, it is termed triple fusion. Since two types of fusions, syngamy and triple fusion, take place in an embryo sac, the phenomenon is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, PC, and develops into the endosperm while the zygote develops into an embryo. Figure 2.13a, fertilized embryo sac showing zygote and primary endosperm nucleus, PEN. 2.4 Post-fertilization, structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruit, are collectively termed post-fertilization events. 2.4.1 Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserved food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the pen undergoes successive nuclear divisions to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm, made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example, pea, groundnut, beans, before seed maturation, or it may persist in the mature seed, e.g. castor and coconut, and be used up during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruit of coconut and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice and maize. B. Figure 2.14a, a typical dicot embryo, B.L.S. Often embryo of grass. 2.4.2 Embryo. Embryo develops at the micropellar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure 2.13 depicts the stages of embryogeny in a dicotyledonous embryo. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo, figure 2.14a, consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyl, which terminates with epimule, or stem tip. The cylindrical portion below the level of cotyledons is hypocotyl that terminates at its lower end in theoretically or root tip. The root tip is covered with a root cap. Embryos of monocotyledons, figure 2.14b, possess only one cotyledon. In the grass family, the cotyledon is called scutellum that situated towards one side, lateral, of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleoryza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyl. Epicotyl has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe the various parts of the embryo and the seed. 2.4.3 Seed. In angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coats, cotyledons and an embryo axis. The cotyledons, figure 2.15a, of the embryo are simple structures, generally thick and swollen due to storage of food reserves, as in legumes. Mature seeds may be non-albuminous or exalbuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. Example, pea, groundnut. Albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. Example, wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucellus are also persistent. This residual, persistent nucellus is theperosperm. Integuments of ovules harden as tough protective seed coats, figure 2.15a. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. As ovules mature into seeds, the ovary develops into a fruit, i.e., the transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. The fruits may be fleshy as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard, etc. Many fruits have evolved mechanisms for dispersal of seeds. Recall the classification of fruits and their dispersal mechanisms that you have studied in an earlier class. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits, figure 2.15b. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of the species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Figure 2.15, A. Structure of some seeds. B. False fruits of apple and strawberry. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats, and help the species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variations. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seeds, or in the presence of seeds which germinate straight away soon after formation and cannot be stored? How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, the seeds lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of a lupin, Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is of the date palm, Phoenix dactylifera, discovered during the archaeological excavation at King Herod's palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar is the case in fruits of some parasitic species such as Orobanche and Striga. Have you seen a tiny seed of ficus? How large is the tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? 2.5 Apomixis and Polyembryony. Although seeds, in general, are the products of fertilization, a few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism, to produce seeds without fertilization, called Apomixis. What is fruit production without fertilization called? Thus, Apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into the embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seeds is costly, and hence the cost of hybrid seeds become too expensive for the farmers. If these hybrids are made into apomix, there is no segregation of characters in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. Summary: flowers are the seat of sexual reproduction in angiosperms. In the flower, androecium consisting of stamens represents the male reproductive organs and gynoecium consisting of pistils represents the female reproductive organs. A typical anther is bilobed, dithecous and tetrasporangiate. Pollen grains develop inside the microsporangia. For wall layers, the epidermis, endothecium, middle layers and the tapetum surround the microsporangium. Cells of the sporogenous tissue lying in the center of the microsporangium undergo meiosis, microsporogenesis, to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represents the male gametophytic generation. The pollen grains have a two-layered wall, the outer exene and inner intine. The exene is made up of sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and generative cell, or three cells, a vegetative cell and two male gametes, at the time of shedding. The pistil has three parts: the stigma, style, and the ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integuments, and an opening called micropyle. The central tissue is the nucellus in which the archosporium differentiates. A cell of the archosporium, the megaspore mother cell, divides meiotically, and one of the megaspores forms the embryo sac, the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate. At the micropular end is the egg apparatus consisting of 2 synergids and an egg cell. At the chalazial end are 3 antipodals. At the center is a large central cell with 2 polar nuclei. Pollination is the mechanism to transfer pollen grains from the anther to the stigma. Pollinating agents are either abiotic, wind and water, or biotic, animals. Pollen-pistil interaction involves all events from the landing of pollen grains on the stigma until the pollen tube enters the embryo sac, when the pollen is compatible, or pollen inhibition, when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma, and the resulting pollen tube grow through the style, enter the ovules, and finally discharges 2 male gametes in one of the synergids. Angiosperms exhibit double fertilization because 2 fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. Zygote develops into the embryo, and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo, globular, and heart-shaped stages before maturation. Mature dicotyledonous embryo has 2 cotyledons and an embryonal axis with epicotyl and hypocotyl. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony.
Don't forget to like and subscribe.